Welcome to Science and Social Tube channel. Kindly do not forget to subscribe our channel by hitting the below subscribe button and press the bell icon.
Friends, today we will learn how to find a new gene. So, for finding a new gene, first we search NCBI database. So here we type NCBI database. This is the interface of NCBI database. Here we directly can search a gene as like scrolling a bar and select the same title as right to written as gene. So here we select a gene and then we type just name as like to and go for finding GRF gene. So I only write the name of GRF gene. After scrolling, then we can saw there are the many genes are available. So GRF gene, lot of genes are available, but here we not decide that which organism we are finding GRF gene. So here you can also write as like to GRF gene in C mice. So here we write the organism name such as plant name. So here directly we can search GRF gene. Here's like transcriptional factor. Here you can see the organism as like to plant name is Zymyce. So this is the sequence of our gene. Here go for BLAST. You can also BLAST to search the occurrence of these genes. Then you can also show here is the gene ID, gene name. So here is the gene description as like to GRF gene is a whole family. So you can also find many genes as compare as linked with the GRF family. You can here see GRF family as like to GRF family 2. You can also see uncharacterized. You can also see GRF 3. You can also see GRF 10. You can also see GRF 1, 2, 3, 4 and so many present in a organism. Here you can also find the description. You can also find the location of the gene as far where is gene presented. Like here chromosome 7, gene is present on chromosome 7, gene present on chromosome 7, gene present on chromosome 5, chromosome 4, chromosome 5 and also location are available. Analysis as like to gene having a different name. Different researcher mention in their paper. So these genes are also important because different scientists call with different name and write their name in this name. So this is called analysis of this gene. So here we can find gene. So then we blast this gene. After the blasting, we can also search FASTA format of these genes. And otherwise, you can directly search FASTA format. Otherwise, if you want to search this gene, then directly search on BLAST and BLAST will find your gene matching with the other gene. This is the option for BLAST. Otherwise, if you want to download the sequence of genes, then you must go back and select the respective gene which are most interested for you. Then as like I am interested in with gene 10, so I select the growth regulating factor 10. I click on growth regulatory factor. So here show this gene directly. Here is the gene growth regulatory factor 10 in organism. This is the organism name. So this is the symbols of the gene. Here is the symbol, here is the description of the gene, here is the tag analysis of the gene, here is the type, gene is the coding type. So this is the other lineage, this gene linked with the different lineage as like to this is the species background which are most important associated with phylogenetic tree. So this gene other name is GRF factor 10 because this gene is also matched with GRF factor is 13. This gene is also matched with GRF factor 13. Friends, you can also see the genomic region as like to genomic contrast. This gene is present on chromosome 5 and the exon count is chromosome exon count is 3. So you can also find the FASTA format of these genes, as like we are going to find the FASTA format. Just you are scrolling down and click on FASTA. Then the next window will also pair this gene in a FASTA format. This is the sequence of this gene. So little waiting, as like to there is the sequence of this gene. You can see this is the sequence of this gene. Here we can copy and save anywhere. Otherwise, if you want to check the similarity of the genes with other organism, you can run directly here BLAST. So when you run BLAST, then this window will be appeared. You can also see the Rodiant database as like to nucleotide collection. Here you can set randomly, and after that, when your accession number of the presented gene is allowed, then the job title will automatically be given. So here you can see the automatically title of this job. So we scroll down, there is the more parametric different settings. You can also set these and click on BLAST. Then here we click on BLAST. The window will be appear in the next form. So please wait. Here searching different similar genes. So good, congratulations. Here we have two different types of the genes are available. Here you can see this is the most important for checking the similarity of the gene. Here you can see these all are organism. These all are organism these genes most similar with. So you can see first two are most important, as like to predictive CMI growth regulatory factor 10 and CMIS mRNA sequencing, because these have to highly matched. E value is 00, Curie coverage is 86-69, total score is the maximum. So you can also search these genes have the accession number. If we click on accession number will show their sequencing, as like you can see here CMIS growth regulatory factor 10 mRNA sequencing. You can also note that here is its locus ID. Here is the locus ID. Here you can see the base pair sequence length is a type of mRNA. So you can also see the protein of this gene. If you want to find the protein of this gene, you can go as like to this is the gene. This is the ID of this gene which we are searching, and this is the protein ID. If you click on protein ID, as like to I open with a new window, so you can also check this is the protein sequence. So you can check growth regulatory factor 10, which we are starting to find, as like its nucleotide sequence is a 2 to 1 amino acid. So you can also see the locus ID of this amino acid is its protein. So then if we want to find the CDS, then you can also go to find the CDS, as like to CDS available is there. Then today we learn how to search a gene, how this gene is used to find the most similar genes in different organisms, and how we can search the CDS, how we can search the gene, how we can search the gene location, how we can search the different CDS have the different base pair. So today we complete the lecture, and hopefully if you enjoy our lecture, then please subscribe our channel and share our video. And if you have any misunderstanding, you can also comment and we can describe for you. Thank you for watching this video.